Hi students, welcome to topic 5. Topic 5 is on Introduction to One Health. This is the last topic for this subject. Let's dive into the intended learning outcomes. So in this topic, we're going to look into the One Health concept. Basically, we want to describe emerging diseases, all right, specifically emerging infectious diseases, as well as re-emerging infectious diseases and zoonotic diseases. We want to list the factors leading to the emergence of all these infectious diseases. Okay, we're also going to look into how an environment affects animal health, including both wild and domesticated animals, right, as well as describing how zoonosis transmission to humans can occur from domestic and wild animals, and how they can be prevented and controlled through the One Health concept. We're going to talk about a little bit about the One Health framework in Singapore and how it helps to prevent and mitigate zoonosis and foodborne illnesses. We're also going to look at the concept from farm to fork to ensure food safety for us to consume as consumers in Singapore. All right, and we're also going to look into the origin of microbial antibiotic resistance and how it impacts to human health. Okay, and finally, we want to look into how the development of antibiotic resistance in microorganisms can be prevented and controlled. So take a moment now to look at the screen. What you're seeing is the SARS-CoV-2 virus. This is the virus that causes COVID-19. Very tiny, invisible to the naked eye. We know viruses are small in terms of its scale relative to the bacteria and other eukaryotic cells. But yet this little virus changed the world, right? As we know back in 2019, 2020, right? So this virus is one of the part of groups of microorganisms that we have learned so far in basic microbiology. These have actually closed international borders, right? Overwhelmed hospitals, disrupted our education, it shaped and even changed our life now, actually. So today I'm using this virus as a starting point to introduce an important concept that I wish to put across, and that's called the One Health concept. Right, so to this end in our subject, we have looked at various group of microorganisms, including viruses like them, right? We know its characteristics and how to culture them, and some of it are often, in fact, starting off from embryonic eggs, egg whites, to culture them. So from the past research, we also know where diseases often arises, right? And we are increasingly seeing diseases arising due to zoonosis and as a result of environmental climate changes, right? So recently in the news, you see some cases of mpox or monkeypox coming in as imported cases to Singapore. So in this case, COVID-19 taught us that diseases do not exist in isolation. They involve humans, animals, and the environment. Right? So what's One Health? One Health is the idea that the health of human, animals, and environment are closely connected, and we're going to look into more details here. You cannot protect one without protecting the other. When one part of it is affected, the entire system is impacted from there. All right. So COVID-19 arises as well as other increasingly re-emergence of infectious diseases. It's linked to animals, influenced by environmental conditions such as urbanization, and also further amplified by our human activity, such as global travel. So this pandemic COVID-19 occur because all these systems, the human activity, the environmental activities, as well as the animal health intersect together. Okay, so how this matters to One Health? All right, let's go into some details into it. So I mentioned just now we are way past the COVID-19 pandemic. Hopefully we won't see another pandemic again. But now there are many cases of monkeypox and the re-emergence of many other diseases, including the Zika virus as well as the Nipah virus, as we've seen in recent newspaper current affair articles. The first cases of COVID-19 is actually detected as atypical pneumonia back in China in Wuhan late many years ago. So before that, nobody even know that this is the SARS coronavirus that was actually discovered, and hence it was new. Zika virus also, it was first discovered back in 1947. It's able to cause many diseases, including the development of development delays in babies when pregnant mothers are infected with Zika virus. They are spread through mosquitoes. Zika epidemic has occurred worldwide in patches. And sometimes, I think back in many years ago in Singapore, we have a similar incident as well. So these are all examples of emerging infectious diseases. And infectious diseases can be defined when one whose incident in humans have increased over the past 2 years, or not 2 years, but 2 decades, all right, or threaten to increase in the future. This is defined as emerging infectious diseases, or emerging infectious disease. So similar to COVID-19, it can be a disease that's totally new to science, or it may be an existing disease that has rapidly increased in incidence, or in a geographical region that have never or had newly appeared from there. All right. So just like the Zika virus back in 2015, when he has a similar epidemic in Brazil, this is considered as an emerging infectious disease. All right. So sometimes a disease can be under control, right? Previously, we have done well with many diseases, including polio, but some become more widespread and prevalent, right? One example is like measles, right? Used to have very low incident rate in developed countries like us and including in Australia, but until recently, the incident went up. Yeah, right. It went up significantly, especially in the US and Europe, and in Asia countries, even in fact in Singapore, we see a few cases, in fact, more than 10 cases last year, right? So, not sure what's the main reasons behind it, but diseases such as measles can be described as re-emerging infectious disease, and this is defined, and it's quite worthwhile to take note that at least 75% of all these emerging infectious diseases are zoonotic in nature. That means there is a transmission of animal disease to human, leading to infection. So you need to know what's the definition of the emerging disease, emerging infectious disease here, all right, as well as the definition of re-emerging infectious disease here. And it is defined as diseases that used to be under control, but whose incidence in humans is increasing recently. Yeah? It will be good to know also what is zoonosis as well as what we have defined here. Okay, so let's look at some of the factors leading to emergence of infectious diseases. Some of the factors include ecological changes and agricultural development. These have lead to some of the emergence of infectious diseases as we come to know about. Like for example, in climate change, the increase in global temperature that had lead to a much more increase in the population of mosquitoes. And recently in Singapore, we have adopted some measures to counter this, but the effectiveness that needs to be studied in the longer term. All right, changes in human demographics are due to migration or even behavior. Global travel also contributes to this. This all overall leads to emergence of infectious diseases as well, right? We saw in the case of COVID-19, right? International travel, as we mentioned, microbial adaptation and changes, right? Some of the changes, adaptation by microbial include AMR, antibiotic or antimicrobial resistance. This is a crisis actually, right? It's an increasingly pressing issue, right? And with all this emergence of the AMR and antimicrobial resistance by microbes, increasingly for the treatment of patients, we can't use certain antibiotics to treat patients, right? And often patients with sepsis or certain infection have to suffer more. They have to stay in the hospital longer, and alternative treatment needs to be arranged to treat them, right? If not, very deleterious effect will happen next, including organ shutdown and so on and so forth. So AMR is a real issue, and we're going to look into that in further detail in the coming slides, right? A breakdown of public health measures and deficiency in the public health infrastructure. This is important also as a factor because all this breaking down of the measures and policies, especially if, for example, in measles, if you were to take away the policy of mandatory or required vaccinations of children when they are young, then this could actually lead to emergence of measles back into the population as well. Causing widespread infection, and we know how deleterious the effects are for measles infections if you don't take your MMR vaccinations. Yeah. So we saw what is zoonosis earlier. Zoonosis is the transmission of diseases from animals to humans, causing infectious diseases. So zoonotic disease can be transmitted through various methods. One of the ways is through indirect contact through the environment. Another way is through direct contact with infected animals. All right. Consumption of contaminated meat products is also possible, leading to zoonosis, as well as through a vector. All right. So through indirect contact through the animal, this can occur when the pathogens from the infected animal, contaminate soil, water surfaces, or plants where we humans will touch or ingest. Contaminated materials as well, airborne particle aerosolize, contaminated food may also be possible. So if you are going to visit, let's say, a certain city where they have a lot of livestock, farming, or even it's a market that contains livestock for sale, then you may have indirect contact with this animal, whether you are contacting it through their cages that they're being contained in, or the infected animals, they may sneeze, and their bodily fluids may come in contact with you, leading to the spread, right? These are some of the factors that can mm, transmit zoonotic diseases, and let's jump into the individual factors here in more detail. So let's look at how indirect contact through the environment can lead to zoonosis diseases. So pathogen transmission at the livestock interface is very complex driven, right? It's particularly driven through environmental contamination and shared resources, often because of our human activity, also because of agriculture or even climate change, right? So the World Organization for Animal Health, WOAH, have mentioned about this in its report, where the indirect contact through the environment usually involves an intermediary vehicle or vector carrying the pathogen itself. So this could be a water body, a vegetation that can harbor the pathogens, leading to the spread of the infectious disease. So we also had a incidents or many cases of hand, foot, and mouth disease, right? Actually, hand, foot, and mouth disease, they are also initially arising from the farm animals or livestock. Pathogens can be circulating between related species here in the first point here, right? The circulation, it can be particularly frequent between the related species, be it pigs or other animals, right? Due to their highly similarity in terms of their physiology and susceptibility. All right, pigs, wild boar, right? These are examples of the hosts that are involved in the African swine fever, right? And they are often transmitted between these two animals, between the wild boar and the pigs, via indirect contact with contaminated carcasses, as what was reported. Okay, so there are also other examples in terms of pathogen circulating between multiple taxonomy orders. So pathogen can actually cross taxonomy barriers, so that means that they are able to infect a wide range of species across multiple orders here. So we are looking at not only mammals, right? Viruses like rabies affecting mammals, including dogs, including bats, but also crossing into other livestock and other animals as well, right? So another good example will be a multi-host pathogen, right? Salmonellosis or Salmonella species. They are known to affect various mammals, birds, even reptiles as well, right? The well-known influenza viruses, avian flu, right? The bird flu in short, right? They affect birds, chickens, aquatic birds as well. Not only confined within poultry, but even humans are affected as well as the disease jump to us, right? So these are just examples of indirect contact through the environment, especially when it comes to bird flu, right? There are humans or even the farmers are handling the cages that are used to contain the chickens as well as the birds. So another factor that can lead to the transmission of zoonotic diseases is the direct contact with infected animals. So when it comes to direct contact with infected animals, this can be in terms of the when you have contact with the infected animals that spreads the pathogens, all right? And this can often be aerosolized pathogen or through bodily fluid exchange, all right? Another factor that can affect the transmission of the zoonotic disease is the consumption of contaminated meat products. So consuming food that is derived from infected animals, all right, can also lead to the transmission of zoonotic diseases. All right, vectors are also a well-known example that can lead to zoonotic or diseases being transmitted or zoonosis. So a good example is the West Nile virus or WNV. So West Nile virus disease is a zoonosis. It's vector borne. It involves the Aedes mosquito, right? So although there are vaccines available for animals, but they are not for human, right? And both this virus itself, similar to the dengue virus, that we have an issue in Singapore here, but although now a bat is under control, right? It belongs to the family of the flaviviral virus family. Yeah. So there are antibodies against West Nile virus that can cross-react with dengue virus. So definitely through a vector, the transmission of the zoonotic West Nile virus disease is actually a possibility. Yeah? So it should be quite obvious by now that we can see the interconnectedness between human health, animal health, and the environmental health are very interrelated to each other, right? All well connected itself. So this is the concept of One Health. So what is One Health? According to the World Organization for Animal Health, the One Health concept summarizes that human health and animal health are interdependent and bound to the health of the ecosystem in which they exist. So we are looking at animals, we are looking at environment, and hence we are looking at humans. So if all of them are healthy, all right, we will contribute to each of the aspects in terms of what the activities that each of these parts are doing. Then these three intersecting part of our world, the environment, the human, and as well as the animals, coexisting with each other. This will help to reduce zoonosis transmission because any changes in the environment can lead to transmission of pathogen to disease, pathogenic diseases to animal, and humans that were not previously exposed to them will be infected as well, right? So if there is a microorganism that's circulating between human hosts and it's possible to jump to animals, that is a very possible real possibility as well. So you can see that One Health, the One Health concept is consisting of these three overlapping areas or areas if you like. So any change of this, any changes made in any of these areas will impact each other, and it happens continuously. Yeah? So I just want to bring across the One Health concept again. This is a collaborative, multidisciplinary approach, all right, that recognizes the interconnectedness of human, animal, and environmental health. The health of human, animals, plants, and the broader environment are closely linked together, and they are interdependent to each other, so as to promote overall well-being and to prevent the disease emergence and spread. So that's the concept of One Health. So the question remains how zoonosis can be prevented and controlled. Right, so we know the human-animal ecosystem interface is very important as part of the One Health concept to safeguard our health or the public health. So direct and indirect human exposure to animal products and to the environment and the ecosystem itself will indefinitely impact the three areas that we come to know in the earlier slides. So collaboration and coordination under the One Health concept is important because this helps to improve prevention and control of zoonotic diseases. Okay, monitor disease threat and care for the health of livestock, as well as to contribute to improving food safety monitoring. So remember when the COVID-19 started, all right, back in 2019, 2020, all right, no one is aware of what it came from, right? People know that it's atypical pneumonia. People didn't know that it was a COVID-19 virus. It wasn't known that it might be a possibility until date also. We don't know whether is it a potential reservoir of this virus coming from bats or even from other animals as well. So if we can work through the One Health concept, involving the different stakeholders here, the different areas that we talk about, then we have a high chance of controlling such zoonosis transmission from humans, especially from domesticated and wild animals. So let's look at the concept of from farm to fork and how in Singapore here measures are being done or even policies are being established to ensure food safety for us to consume. So how foodborne illnesses are prevented and controlled in Singapore? So the concept of from farm to fork begins when the food is being produced and ultimately it landed on our table. That's why the term from farm to fork. Between any of these stages, when the food is being produced at the farm and ultimately the food being presented on the table for consumption, foodborne zoonosis is a real threat. Food contamination can also happen. Foodborne illnesses can also arise throughout if any of this breaks between this whole value chain. So contamination can occur at very different stages of this whole process. Right, so to minimize the risk of food contamination, okay, action is needed at all stages of the food chain, from production all the way to consumption, and hence the term, as I mentioned just now, farm to fork. So prevention, detection, and control of diseases at the primary production phase, that means at the farm area, reduces the risk of human illnesses. When we talk about the different factors that can lead to zoonotic disease transmission. So this includes indirect contact with infected animals as well as direct contact with infected animals. So let's give an example for poultry meat like chicken. Okay, the chicken, they can acquire bacterial such as Salmonella and Campylobacter from the environment as a result of from the environment or from their feed that they've been provided to, resulting in an infection in the chickens themselves. Some of the possible route of infection, route of transmission, especially will be through direct routes. So some of these direct routes include human salmonellosis and Campylobacter cirrhosis arising from consumption of the infected poultry meat. An indirect route for the transmission of the zoonotic diseases can be on the slaughterhouse, where the slaughtered carcasses are contaminated and they are contaminated via contact with the intestinal content of the chicken, and this may inevitably be in contact with the staff who are working at the slaughterhouses. All right, so many of the foodborne pathogens are actually zoonotic. All right, so you have the E. coli 0157 strain. These are actually found in cattle. You have the Salmonella enterotitis that are often found in the poultry and in eggs. Campylobacter jejuni, very famous bacteria that's found in the poultry, as well as Streptococcus elangitea, which is known as a Group B Streptococcus. It's found in fishes. So in our era of globalization now, our food chain has actually grown much more longer and more complex, involving many parties between across this whole value chain. So the International standardization of food production procedure and safety measures is hence of utmost importance. So the World Organization of Animal Health, it collaborated with other international organizations as well, including WHO, and developed international standards for food safety to ensure that the whole food production chain is appropriately regulated. So veterinarians also in the different countries worldwide also carry out wide activities in animal production systems that contributes to ensuring the production of safe food. So, so why are vet services are important? Because they are ultimately also responsible for controlling pathogens in animals. Okay, but also importantly, they need to work with other stakeholders involved in the food system, including the feed producer, the farmers, the processors, the wholesaler, the middlemen, the distributors, and so on and so forth. So in summary, I hope that I have impressed upon you the One Health concept, the concept that captures the interrelatedness between humans, animals, and environment. Right. So in summary here, we have defined what is emerging infectious diseases, re-emerging infectious diseases, and zoonotic diseases. Right. Factors leading to emergence of infectious disease. One of the factors include indirect contact, direct contact infected animal, consumption of contaminated meat products, as well as through vectors. As we look into some of the examples, including the West Nile virus and the dengue viruses. We also look at how the environment affects the animal health, including both wild and domestic animals. And we know that what human activities are one of the main culprit that had led to certain spillover events. When we talk about spillover events, we talk about microbes, including viruses that infect animals, jumping from species, and then eventually becoming transmissible between humans. All right. And the last point here on how zoonosis and foodborne illnesses are prevented and controlled under the One Health concept. Remember, we talk about the three different areas of cooperativeness, right? Interrelatedness. So it's important that we embrace the concept of One Health so that we can work together to actually reduce the incident of zoonosis and foodborne illnesses. So this concludes part 1 of topic 5. I will see you in the next video. Thank you. Hi students, welcome to part 2 of topic 5. Topic 5 is focused on introduction to One Health. In part 2 of this e-lecture, we will be looking at microbial antibiotic resistance and how it impacts to human health. We're going to talk about some of the development of AMR or antimicrobial resistance in microbes and how they can be prevented and controlled in the upcoming parts of this video. So for AMR is known as antimicrobial resistance. This is where the microbes change in a way that renders medications such as antibiotics ineffective. They become resistant to most drugs and hence they are often labeled as superbugs. In the history of antibiotics, all right, back in 1928, Alexander Fleming was a great scientist who noticed a fungal contaminant could actually inhibit bacterial growth. All right, and this fungal contaminant eventually came out to be marketed as penicillin, and clinical trials were successful to treat patients for blood poisoning or sepsis. Right, and he won a Nobel Prize for that. Okay, but in 1940s there were signs of antibiotic resistance. Right, in fact, E. coli actually produces a substance in counter to this penicillin, right? They produce penicillinase, right? Sounds like enzyme that actually degrades penicillin, rendering it inactive, right? Further research back in 1960s, I think there was a discovery of methicillin, a different form of antibiotic. This helps to slow down the penicillin resistance spread. However, 20 years later, there was resistance to methicillin and this became endemic in the US, right, causing a huge number of hospitalized patients to be down or infected with S. aureus, right? Often these are all nosocomial infection, right? In i.e. the infection happening in the hospitals. So how big is the problem for antimicrobial resistance? So the WHO estimate by 2050, antimicrobial resistance is estimated to be one of the top global public health threats that will contribute to more than 10 million global deaths and trillions of dollars in economic loss worldwide every year if no action is taken. So far exceeding the death caused by cancer, which estimated to be around 8 million people a year. So this is a very big problem, and increasingly we are seeing resistance in antimicrobial resistance in microbes, and in Singapore here, actually, and in fact worldwide, people are taking steps to try to curb this issue. So I want to talk about how antimicrobial resistance can come about in bacteria and leading to the transfer of such resistance between its population. So we are looking at mutations, genetic mutations that can lead to antibiotic resistance. These mutations are often spontaneous, natural mutations of one bacterium itself. So in the presence of, or in fact, if in the absence of antibiotics, the bacterium itself may spontaneously have AMR antibiotic resistance, as indicated here, shaded in gold, all right, or dark yellow. So in the presence of antibiotics, these the antibiotic sensitive bacteria will die, right, due to its action, right, allowing the resistant bacteria to survive and reproduce. All right, this is in the case where your antibiotics are present. Okay, those that are sensitive to them will die off, leaving only the resistant bacterium, right? In the living bacterium themselves, they are resistant towards the antibiotic. So there's a continuous presence of the antibiotic resistance. This ensures the resistant bacteria survive over time, and with selective pressure, right? These are the forces that causes the organism, in fact, in this case, the bacteria, to evolve to a certain state, right? In the absence of antibiotic, this antibiotic resistant bacteria continue to thrive, all right, even in the absence and after many generation, all right, they are able to form many generation, and a new population of all these microbes emerges that are resistant towards the antibiotic use. All right, so one of the ways that bacteria can assume antibiotic resistance is through such mutations. So there's antimicrobial resistance, partially due to horizontal gene transfer. Horizontal gene transfer allows bacteria to exchange their genetic materials, especially the antimicrobial resistant genes. They are found on plasmids as well as on DNA sequences. So the genes themselves are found in plasmids. Plasmids are actually circular DNA that's independent of the bacterial chromosomes or transposons. All right. Transposons are actually DNA sequences that can be mobilized by the bacteria or cell, and they can jump between DNA sequences themselves. So the genes may be carried by plasmid or on transposons, also known as mobile genetic elements. So for horizontal gene transfer for antimicrobial resistance are often carried out, in fact, through different processes. Some of these processes includes conjugation, transformation, as well as transduction. So let's have a look at the individual horizontal gene transfer methods here. Okay, so we mentioned about conjugation as the way of bacteria spreading AMR genes. So conjugation is the transfer of DNA directly through cell-to-cell contact, often involving the plasmids. This is the most common method of horizontal gene transfer. Bacterial to bacterial cell contact can be established through the pillars, which is an appendage on the bacteria itself, to transfer, or in fact to transfer the plasmid across two bacterial cells. Another way for horizontal gene transfer to happen is through transformation. Transformation involves picking up DNA directly from the environment and inserting into their own genome. If you can imagine, in a population where there's a lot of lysis of dead bacteria, you got a lot of plasmids that are floating in the environment, right? So some of these DNA plasmids can be picked up by living bacteria within this population, and inserting it into its own genome. Another method will be through transduction. Transduction is the transfer of DNA from one cell to another by a virus, right? This virus in this case is called bacteriophage, right? And this bacteriophage is responsible for carrying the genetic elements, all right, into or in fact between bacteria as it infects bacteria. And finally, mutations is also one of the ways that antimicrobial resistance can arise, as we see earlier. Genetic mutations in response to selective pressure leads to the formation of the mutated genes, resulting in the AMR, antimicrobial resistant genes, which will eventually be transferred within the population through these three methods as we have highlighted here: conjugation, transformation, and transduction. So just to recap, how antimicrobial resistance arises. When we use antibiotics, some bacteria die, as we see in earlier slide. However, the resistant bacteria can survive and even multiply in the presence of antibiotics as it is resistant to it, and i.e. it has the antimicrobial resistant genes. So the more we use antibiotics, the more chances the bacteria will become resistant to them. Antibiotic will not work eventually, okay, and if we need them in the future next time, then we will be in trouble. Now we know that the antibiotics or even the antimicrobial use in medicine is widespread. It's therapeutic. It's used for treatment of diseases, especially for sepsis. In terms of its growth promotion, back in 1940s, as we have seen earlier, right, there are reports of antibiotic resistance already, right? During that time, antibiotics was also used to enhance growth in chicken, with sub-therapeutic levels of antibiotics given to animals to enhance growth. But since 2006, countries worldwide have already banned the use of antibiotics due to the rise of antimicrobial resistance. Such antimicrobial use in animals are often used as prophylaxis, right? So prophylaxis is actually treatment to the animals that are not currently ill, but just as a preventive measure, just in case, so-called, because these animals are at a high risk of infection. So often vets will often give antibiotics as a prophylaxis. Even the farmers do as well. Metaphylaxis, this is defined as providing treatment to a herd of flocks if they are extremely at risk of being in an outbreak due to their exposure to certain disease agent or due to unfavorable environmental conditions. So in the metaphylaxis stage, this is where the vets will actually provide treatment to the animal herd of flocks that have a high risk of coming out of the outbreak. Nonetheless, there we should encourage prudent use of antimicrobial antibiotics, judicious use as well as leading an antimicrobial stewardship. And currently now worldwide, we are fast approaching this approach because this helps in the optimal selection of drugs, dose, and duration of antimicrobial treatment, along with the reduction of any inappropriate excessive use of antimicrobials. Remember that the more we use them, right, there is a higher chance that the microbes will become resistant to them. So in summary, we have talked about the origin of microbial antibiotic resistance and how it impacts to human health. Right. So evolutionally, and these AMR genes are transmitted through horizontal gene transfer between bacteria. Here we are looking at methods such as conjugation, transformation, and transduction. All right. How the antibiotic resistance came about? Often these are spontaneous natural mutation that happen. Right? And we also look at how the development of antibiotic resistance in microorganisms can be prevented and controlled. And often this will depend on the usage of antimicrobial through prophylaxis, through metaphylaxis, and of course with prudent use of these antibiotics for animals. So this concludes part 2 of topic 5. I will see you in the next video. Thank you. Hi students, welcome to topic 5. This is part 3 of the topic 5 e-lectures, Introduction to One Health. In this last part of topic 5, we'll be looking at describing how the development of antibiotic resistance in microorganisms can be prevented and controlled. So I want to talk about some of the potential antimicrobial resistant pathways that could happen in Singapore. With human use of antibiotics, the spread of resistant, or in fact antibiotic resistant or antimicrobial resistant bacteria to the community and to the environment is very possible, and this could be done through water bodies. Agricultural use of antibiotics, right, from agricultural farms. Some of them may develop resistant bacteria, especially in the animals, or even in some of the primates. This could actually spread to the environment as well through water bodies, perhaps through their contaminated bodily fluids or fecal matter. The veterinary use of antibiotics in the vet clinics and animal clinic, okay, they may also have developed a chance for forming antimicrobial resistant bacteria, spreading it through the environment as well. Here, we want to note is that the resistant bacteria in the environment itself could possibly spread to the wildlife in addition to humans in terms of the microbes present in the humans. So just want to outline some of these potential pathways that may happen as we embrace the One Health concept here. So the One Health concept was adopted in Singapore as well as to help us to prevent and mitigate zoonosis and foodborne illnesses. Back in many years, there was a National Strategic Action Plan, or AMR. The Antimicrobial Resistant Coordinating Office was established, I think, back in 2017 to 2015 to coordinate Singapore's One Health approach against AMR. Right, this works closely with Singapore Food Agency. They work with MCRO, okay, as well as the Ministry of Health and other bodies in Singapore to prevent antimicrobial resistance across the different sectors, as we know, human, environmental, and animal sector. And often they aim to promote the One Health collaboration between the teams here. So there was actually also a plan, a One Health plan, so as to help to strengthen coordination between these sectors. And these include measures including enhanced monitoring of zoonotic and vector-borne diseases, all right, improve food safety measures, right, coordinated management of antimicrobial resistance, as well as an integrated approach to environmental health protection. So all in all, the point in this slide here is to inform you about the coordination between the sectors and the ministries that aims to reduce the risk of disease transmission to both people, animals, their pets, as well as ensuring the food safety from farm to table. So as I mentioned just now, the National Strategic Action Plan on Antimicrobial Resistance was launched back in 2017. And the team focuses on all these aspects, looking at education, looking at surveillance and risk assessment, research, prevention and control infection, and importantly, the optimization of antimicrobial use. Because excessive use, as we know, can lead to an increased risk of microbes developing resistance towards our antibiotics. So the spread of antibiotic or antimicrobial resistance cannot be underestimated. There are many ways of how antibiotic resistance can spread. And earlier in the slide, we see some potential pathways that could actually lead to such spread of antibiotic resistance. But here are two examples of how this can spread from animal to humans. Right, animal consume antibiotics in their feed and develop antibiotic resistant bacteria in their guts. Right, human consume the animal meat, and if it's not cooked properly, it may contain the antibiotic resistant bacteria, right, resulting in the spread of the antibiotic resistant bacteria to humans. All right, so this is one way. Another way could be from humans to humans. Right, Mr. X may be taking a antibiotic and develop anti-resistant bacteria in his gut. He stays at home in contact with the general community, and he may spread the antibiotic resistant bacteria to the community as well. Right, earlier we saw there are also possibility for contaminated fecal matter to be spreading the antibiotic resistant bacteria through the environment, possibly through the water as well. So these are some examples of how the spread of antibiotic resistance can happen. So in order to curb and control such antibiotic resistance from developing, right? The Global Action Plan on antimicrobial resistance was launched back in 2015 by WHO, the World Health Organization, right? So they aim to improve awareness and understanding by raising public knowledge and as well as professional knowledge in terms of encouraging behavioral changes, for example, right? Training physicians know in terms of prescribing antibiotics, as well as communication and education to patients to finishing their prescription of antibiotics and avoiding self-medication, right? These are part of the five strategic objectives that WHO have against antimicrobial resistance from developing. All right. The second point in terms of strengthening knowledge through surveillance and research, right, is by monitoring certain patterns and also consumptions of antimicrobial drugs through surveillance systems established in various other countries, right? So one of these system they call the Global Antimicrobial Resistance and Use Surveillance System, or GLASS for short. Right? You don't need to know this, but this is just for your information. All right. The third strategic objective is to reduce the incidence of infection, right? So prioritizing education through hygiene, improve hygiene, infectious control, prevention, right? Some of these measures both in the healthcare setting and also in the community setting, so that the aim is to lower the overall need for antimicrobials, right? The fourth strategic objective is to optimize the use of antimicrobial agents. Okay, so judicious use of antimicrobial, use only when necessary, at the correct dose, at the appropriate duration, at the appropriate time. All right. The last objective is to develop the economic case for sustainable investment that takes into account the needs of all countries and increase investment in new medicine, diagnostic tools, and vaccine, and so on and so forth. Right. So in this case, the objective here is to actually ensure that all countries, regardless of their resource level, especially low-income countries or underdeveloped countries, they still have accessible and affordable and sustainable medicine and diagnostic tool. So all in all, the strategy helps to minimize selection pressure, right, and helps to slow down resistance strain from developing, right. All these strategy work together through the One Health approach, so that you can disrupt the cycle of antimicrobial resistance, right. It also helps to prevent disease transmission as well, right. By one of the objective is to through education, right, through the reducing the incidence of infection here. So by preventing disease transmission, you negate the need for antimicrobial. Okay, and all these can be done through improving hygiene conditions, sanitation condition, and making sure that countries, low income or underdeveloped countries, have readily access and affordable diagnostic tools or medicine. So similar to WHO, the World Organization for Animal Health also has a similar strategy against AMR. So their objective are herefore, right, improving awareness and understanding of AMR, strengthening knowledge through surveillance and research towards AMR, supporting good governance and capacity building, as well as encouraging implementation of international standards. So all in all this helps to minimize selection pressure as well, slowing down resistance strains from happening, right? And their measures work together so that you can preserve the efficacy of this life-saving medication in terms of their antibiotics by reducing the need for these antimicrobials and ensuring judicious or responsible use, right? All in all this helps to prevent disease transmission as well. So you need to know what are some of the strategies outlined by WHO, as well as the World Organization for Animal Health against AMR. So all those in the box here will be important to know how it helps to minimize the selection pressure. So I hope I have impressed upon you, okay, how important it is to control this antimicrobial resistant crisis. It requires a One Health approach, Okay, recognizing that human, animal, and environmental health are interconnected. All right. This way, the development of antibiotic resistant in microorganisms can then be prevented and controlled. So we have discussed in details on how antimicrobial resistance can come about, yeah. However, how do we know which antimicrobial reagent or the antibiotics are showing resistance or susceptibility against the various bacterial microorganisms? So typically in the clinical microbiology lab, we will use a technique known as the antimicrobial susceptibility testing. So this technique is also known as the Kirby-Bowell technique. It is a agar diffusion test which involves using a special plate of media, typically a solid agar, and spread across with the test bacterium, typically using spread plate, and then using different small discs containing a pre-measured dose of the antimicrobial or the antibiotics dispensed on the bacterial lawn itself. So here I'm showing on the right hand side here a representative image. You have a spread plate with a bacterial lawn, and once they are fully confluent, you can dispense and or apply the antibiotic disc on the lawn itself and then incubate it. So these antibiotics could be vancomycin, penicillin, erythromycin, streptomycin, for example. So it's a way of testing a pure culture of the bacterium against the various antimicrobial drugs in vitro. In vitro means we do it in the laboratory. So upon incubation, all right, you can actually determine if there is any zone of growth inhibition. So like for example, in this case here, you have see a zone of clearance around the disc, the antibiotic disc. The area around the antibiotic disc is uncolonized. So and then with this, you can actually use a ruler to measure. Right, for example, for erythromycin, you can use a ruler to measure. This is around 27 millimeters, and then comparing it with a standard or a control for each of the drug to determine the clearance of growth. So you can expect that each antibiotics will have different zones of inhibition size to determine if the microbes is resistant, intermediate, or susceptible or sensitive to the antimicrobial agents here. So the zone, just to take note, is measured from age to age, from the no growth zone straight across the antibiotic disc. So from here, you can see a little marker mark here, and then all the way to this end. This whole area is the zone of growth inhibition. So as mentioned earlier, after measuring the zone of clearance or the inhibition, you can actually compare it to controls or standards to determine if the zone size is corresponding to a drug susceptibility towards the antibiotics or a resistance towards the drug. So here I'm showing a table here for a example result for Staphylococcus aureus, which is the skin commensal bacteria. So if you were to grow them onto a lawn or on a spread plate itself, and then you place or dispense the different drugs containing this on the lawn itself, and then you measure the clearance, all right? And then comparing it to the standards itself, you can see that Staphylococcus aureus is resistant to streptomycin. Okay, around 11 millimeters of zone clearance. Anything that is below 11 millimeters is the zone size required for resistance. For vancomycin, it is sensitive or susceptible, right? The clearance zone is about 15, so anything above 12 is susceptible to the drug, right? And finally, for erythromycin, right, the growth inhibition zone was around 15 millimeters, and this is quite intermediate between 18 to 13. So there's intermediate sensitivity towards erythromycin. So that's how you read out the antimicrobial susceptibility test in the form of an agar disc diffusion method. So in summary, we have went through the One Health framework in Singapore and look at how it helps in preventing and mitigating zoonosis and foodborne illnesses. So there is a national strategic action plan on antimicrobial resistance, as well as the One Health plan. And these plans outline some of the measures that includes enhanced monitoring of zoonotic and vector-borne diseases, improving food safety measures, coordinated efforts, as well as integrated approach to environmental health protection. We know that the One Health concept is important because it entails the three domains or the areas that we look at, which is human health, all right, animal health, as well as environmental health. We also look at how the development of antibiotic resistance in microorganisms can be prevented and controlled. So the WHO has outlined 5 strategic objectives against AMR, okay, as well as the World Organization for Animal Health, its strategies against AMR. Okay, in totality, these strategies actually help to minimize the selection pressure and help to slow down the rate of antimicrobial resistance. All right, and also some of this policy, it helps lower income countries or underdeveloped countries to have readily and affordable access to medical diagnosis and tools and materials that are important in the fight against AMR. Right, and finally we have looked at how we can perform antimicrobial susceptibility testing to determine whether the microbe is susceptible or sensitive to certain drugs, particularly the antimicrobial reagents or the antibiotics, okay, by using a disc diffusion method that involves placements of your antibiotic disc on a bacterial lawn and then measuring out the zone of clearance, zone of growth inhibition, and comparing it to standards or controls to determine whether the bacteria is sensitive, resistant, or have intermediate sensitivity to the antibiotics tested in vitro. All right. Some of a recommended reading towards the end of this slide here. So please do have a read in the recommended text on chapter 25 and the reports that are provided by the respective entities here. Right, they have included a short abstract in the beginning of this report. You will just need to read the abstract to gain an understanding of what is AMR, as well as some of the objectives that we have outlined in our lesson today, and some of the measures that could be done to address, in fact, to control the AMR crisis that we are seeing now. So thank you very much. This concludes topic 5.